Okay, this is our final video where I'm going to teach you about fermentation. So basically, fermentation is what our cells do when there isn't any oxygen. And so, or at least there's an oxygen debt. We don't have enough oxygen to do oxidative phosphorylation, but we still need ATP. So when we're running, for instance, you're doing oxidative phosphorylation already at maximum speed, but you need even more ATP, so you'll do fermentation and oxidative phosphorylation simultaneously. But let's say you don't have the oxygen at all, like if you're drowning or if you've had a heart attack, then all you can do is fermentation, which is only a temporary solution. Yeast, since they're single-celled fungal organisms, they're very simple and they can survive on fermentation alone, and so they do that without any problem. Okay, so in order to understand fermentation, let's quickly go over glycolysis again. Remember, with glycolysis, you start with glucose, which we have C6, H12, O6, and we do glycolysis. So glycolysis is really the first part of fermentation. So remember, glycolysis, we phosphorylated the glucose, we split it in half, and to make 2-pyruvate. But in doing that, we also made 2 NADH, right? And then we made 2 ATP. So the products of glycolysis, remember, are 2-pyruvate, 2 NADH, and 2 ATP. So you can do this and make your 2 ATP, but the problem is if you want to do glycolysis again, you're eventually going to run out of NAD. You're not going to have any empty trucks coming to be able to pick up the hydrogen, so it's going to run to a halt if we can't figure out a way to oxidize this NADH. So it's normally the NADH should be oxidized in the oxidative phosphorylation when the NADH is donating electrons and protons to the electron transport chain. But if oxygen isn't present, right, we're not going to be able to pick up the electrons at the end of the chain. So that's going to run to a halt, and so we'll start having all of our trucks filled with hydrogen, and then we won't be able to do glycolysis. So we basically need another way to oxidize the NADH, and so that's what fermentation is all about. So humans and a lot of bacteria do something called lactic acid fermentation. And so what that does is you take your 2-pyruvate and you do another step to it. You basically oxidize the NADH and you rearrange this into take the hydrogen and you add it to the 2-pyruvate, and you make the pyruvate into molecules of lactic acid. You actually make 2 lactic acid. And so lactic acid is basically a reduced form of pyruvate, and then you end up with your NAD again, and so then you can keep making lactic acid. So the whole purpose of making lactic acid is really to just get rid of the hydrogen on NADH so that you can have NAD again, so you can run glycolysis. So humans and bacteria make lactic acid. This is what builds up in your muscles. This is what makes the acid of fermented foods like kombucha and yogurt and sour cream and things. That's what's making that sour. If you're a yeast, let's go back to our pyruvate. Yeast do a step in which they reduce the pyruvate with hydrogen, but also they remove carbon in the process. Actually, they remove carbon dioxide. So if we remove carbon dioxide from this and we reduce it, we end up with ethanol. So ethanol only has 2 carbons. So you produce carbon dioxide in ethanol fermentation. That's the bubbles that yeast produce. And so you make ethanol and carbon dioxide. So this is the alcohol of alcoholic beverages, the bubbles that you would get if you close the container. So like beer and wine, if you could keep the container closed, you'll also get the CO2 to carbonate it. And so that's, instead of lactic acid fermentation, that's alcohol fermentation. Eventually the ethanol gets to be so strong that if it's in a closed container, it'll kill the yeast. So you can't really make more than about, I think it's like 6% ethanol without just stopping the process, because the yeast will die in the alcohol. So that's fermentation. Okay, that's pretty much it. Oh, the other thing I wanted to talk to you about was the control of cellular respiration. So, and I mainly want to mention this because on the AP test, they're going to be talking about perturbances to systems and how cells regulate and maintain homeostasis. So cellular respiration is highly controlled through a feedback mechanism. I'm going to get back to our glucose here. So C6H12O6. Okay, so the third step of glycolysis is catalyzed by an enzyme called phosphofructokinase, or PFK. And PFK is an enzyme that is regulated. So it is sped up by ADP and inhibited by ATP. So if you remember the third step, so let's go ahead and go through the steps. So the first step of glycolysis, you take ATP and you phosphorylate the glucose. The second, and you make ADP. The second step is you turn this into fructose, so you have fructose 6-phosphate. And then the third step is you phosphorylate the other side of the fructose to make fructose 1,6-diphosphate. It's that reaction right there, that going from fructose 6-phosphate to fructose 1,6-diphosphate, that enzyme is phosphofructokinase. And that enzyme is sped up by ADP. So in other words, if the cell is needing ATP, you're going to want to do cellular respiration more quickly. And so that enzyme can speed up. And then it's inhibited by ATP, because if you already have lots of ATP, you don't need to do cellular respiration, so it slows down. So it's the throttle, so to speak, of the whole thing. Because it's happening in the third reaction of cellular respiration, which is like 40 steps, you're controlling it early on so you don't waste all of the intermediate products of cellular respiration. So if you remember enzyme pathways, you want to have the inhibitor inhibit at the beginning of the enzyme pathway, so you don't end up with all these useless intermediates. Okay, that's it. I know that's a lot. So I'm going to have you guys fill out all the handouts. Hopefully that will help you internalize all of this. Okay.